our next speaker is Dr. Andrew Steinsapir, who's coming to us to speak about novel path to the brain, crossing the blood-brain barrier for gene therapy. As Dr. Wilson alluded to, we don't stop. So it's important as we advance genetic medicines for Angelman syndrome, we don't just stop and say, well, this is it. If this doesn't work, nothing else will. We have to ensure that we have options coming behind. For every version 1, there's a version 2, and for every version 2, there's a version 3. And as we think about that and we think about the transformation of science, we have to keep up with the science. And this is something that still is in somewhat early stages. We don't have evidence or data really in humans as it relates to these new type of capsids, but it is what's upcoming, and we are going to see over the coming years a lot of new activity in this space. And so I invited Andrew to come and speak to us about the incredible work that him and his team have been doing at Apertura, who have been a really great team engaging and embracing rare disease parents and families and organizations to ensure that all advocacy groups have access. They have access to new technology, and money and time and availability does not become their barrier. And so Andrew has really been quite impressive as it relates to his communication and engagement with communities, particularly this one. So Andrew, please.
Thank you so much, Allison, and it's truly an honor to get to be here, and especially following a very tough set of acts. As Allison noted, my name is Andrew Steinsapir. I help lead Apertura Gene Therapy. I have a day job. I work at a company called Deerfield Management. We do healthcare investing. Our motto is advancing healthcare, and I've always tried to add the coda on to that of sometimes one patient at a time. What we did is help start a company called Apertura Gene Therapy, and we said we know the problems because the leaders in the field have been calling them out for years. We need to understand how to build tools, in this case AAV, that will work, that can move forward to make life easier for what we're doing. And what we wanted to do is start with that end in mind, not the first patient that gets a drug, but rather the 1,000th patient in commercial that gets that drug because it's already been approved. So what we did was we said we want to use the science that we understand, what we've learned in antibodies, what we've learned in other drugs, and start with the receptors, the things that we target, the things that help us get to where we're going, and leverage those. We are standing on the shoulders of giants, including our scientific founder, Ben Deverman, who has started all of this work and is the reason why we've moved this forward, not just on the science, but also in the purpose of making sure that everyone has access to what we have, because we alone cannot do everything. It is only when we are together that we are the smartest and the most capable. So with that, and I think we had some explanations of what AAV is, and I was told very specifically by Allison that the people in this audience know more about science than I ever will, but nonetheless, I will make sure to have this. I've always thought about AAV as helping give a software update. As it was noted earlier, we have viral genes that we can take out, that's the stuff on the left, and instead what we put in is on the right. It's that new set of instructions. And AAV is really good at going into cells and dropping off those instructions so that they can be read. Those instructions could be things like how to make UBE3A. They could be instructions like how to do gene editing or other things. And because we're able to drop it off and it can hang out and be in the cell, we don't have to keep redosing this. Now with that, we have all of these opportunities that we can address. So the question is, why aren't we already doing it? Part of the reason why we're not doing it is the things noted by Jim and by Liz in the complexity of moving these forward, because certain AAVs are better at different things. And frankly, AAV that exists in nature, which has been around for billions of years and has never caused disease, isn't really meant for gene therapy. It's meant to be what it is. Nonetheless, we've been able to find through so much work and energy and time which ones of these naturally occurring AAVs can be used to get to certain tissues, be those the central nervous system or the liver or the eye or the muscle. But really what we have been doing is working with research tools and trying to find the right ones that maybe meet our needs. And with that, we need that progress. It's important that we continue that work and move it forward. That's why there are drugs that are approved. That's why there are more than 4,500 babies that have received Zolgensma for SMA1. These things have changed the world, and they will continue to do so. What we're hoping to do is help expand that opportunity by building new capsids with new capabilities that can help across more diseases and make it easier to administer those products and be able to do it at lower doses for all of the reasons that folks were talking about earlier. And in a lot of ways, I think that we've gotten there, at least in our early research. The work that I'm showing to you right now is a mouse brain on the left in blue, and then on the right is another mouse brain, but one that received our first generation capsid that uses the human transferrin receptor to cross the blood-brain barrier. I'll be telling you more about that later, but the thing I want you to see here is all those little green dots. All those little green dots are software updates. Those are cells that received that virus. And if I had the other slides that I had in here, but I have a limit on the amount of time I have, I would turn off the blue and you would see just a green brain on that side. And the reason that we're able to do that is because we're using biology the way that it was meant for. We're leveraging a receptor called the transferrin receptor. The transferrin receptor is named that way because scientists are not creative. The transferrin receptor binds to transferrin, hence its name. What the transferrin receptor does normally is help the body by grabbing on to transferrin, which holds onto iron, and move it across the blood-brain barrier. Iron on its own hanging out would be actually toxic, so our bodies put it into this protein to help protect us. Now, that protein, unfortunately, is too big to get in the brain, and that's where we actually need a lot of iron to make energy. The same way that people say that if you do a crossword puzzle, you don't have to run a mile because you're burning a lot of calories, although jokes aside, I know doctors, I still should be running a mile. We need to get it in there. So we're leveraging the same receptor that's normal job is to help be a ferry to bring things across, to bring our virus across. And in fact, this receptor has been used across hundreds of patients, both pediatric patients as well as geriatric patients, to help move other biomolecules across. You heard Dr. Wilson talk about Hunter syndrome a minute ago. There is a drug that is approved that uses the human transferrin receptor to bring across the medicine for Hunter syndrome for folks, and that drug is approved in Japan. And there are other things that are moving forward from both JCR, Denali, and Roche in the US to do the same thing. Now, all of those are doing biologics, those like an enzyme. What we're doing instead is saying we're going to use our virus to do that because it also uses the same receptor. Now, with that, we think that that means that we have an opportunity to help across a number of disorders. And of course, Angelman is on there, but it's more than just that. It's all of these diseases that we haven't heard of yet, the ones that have 5 letters and 2 numbers that maybe 3 people in academia are researching. We think that we have an opportunity to make sure that those folks have access to this too, and that they can build upon this platform, like Jim was noting, in being able to consistently be able to move forward because we're using the same tools to do the same job over and over again. For our work, we are progressing actively with multiple rare disease groups. We have one program that should be in human trials at the end of 2026. We have another that should be in human clinical trials at the beginning of 2027. That's exciting, and honestly, talking to all the folks in that community and others, I know it's never going to be fast enough. But the reason it's taken this amount of time is because we're making sure that we're running the right experiments at the right time in the right order, that we are not cutting corners, but moving forward correctly. With that, and even though we have oodles of safety data in engaging with the human transferrin receptor for other drugs, it's never been done with a virus, which is why it's been important that we've executed critical studies in non-human primates and in mice to help understand how this medicine can work. We're excited for this to move forward, and it's really a great pleasure for me and a privilege to say that our first partnership was not with a therapeutics company, but rather with the Rett Syndrome Research Trust, an organization like FAST that's devoted to working for their patient advocacy community to progress things. We've also been able to work with a number of other groups, and our goal is to actually make sure that this is available across the board for folks in academia developing products, as well as people in companies and rare advocacy groups. And with that, it wouldn't be a science talk if I didn't show you data. You saw brains earlier, and this is the part that I'm most excited about, outside of answering your questions after this, because we have a poster. But we are able to use dose levels here that are 50 times lower than what are used in the currently approved therapies for intravenous administration. And even when at that super duper low dose level, we're reaching almost all of the cells in the brain. What you see here is AV9, which doesn't work really IV very efficiently in adult mice. Then you see a capsid that only works in mice, which it's not going to do anything for people. And then you see our capsid, TFR1 CapX, that uses that human transferrin receptor to cross that blood-brain barrier. And to that point of looking at other tissues and how that virus goes to different places. The thing that's so exciting for us, and that you can see here in that pink magenta, is that at these doses, we're getting about the same amount of virus into the brain, into the CNS, as we are getting into the liver, bringing it into parity, rather than what we've seen with other standard capsids in this first generation of technologies, where that ratio is 100 to 1 or 1,000 to 1. What that means as we think about this is that not only are we able to address this with a much lower dose, but the places that we're going are closer to where we want them to be for both neurological disorders and neurometabolic disorders. What we've done here is not just to say we're going to build a tool and then not use it. We've also been progressing internal programs, we've been collaborating with others, and we recognize that it is not just up to us to move these things forward. The first thing that we want to do in every case is to sit down with the patient advocacy communities and understand what the needs are, because the stakeholders in the room are the people that are going to actually use those drugs. And that's why it's important that it's everyone in here that feels comfortable with the data that is generated, because it's not just what the FDA says okay to, it's not just what the doctors say okay to, it's also understanding these things. And it's wonderful to see such great presentations explaining everything like you saw earlier, so that everyone understands what's moving forward. Nonetheless, with that, in our programs that we've been moving forward for TSC, another genetic disorder, as well as for prion disease with Sonia Valeb, we have already gotten alignment on those programs with the FDA for what our path forward is. And that's a path forward where we can use these mice that have the human transferrin receptor knocked into them and use them as the direct model for human dosing, because it has the exact same receptor and that drug interacts exactly in the same way and helps us have predictable dose levels across the board. With that, we've also been able to help make sure that the data that already exists, the research that's already been done, doesn't need to be done over again, and instead we can continue to leverage those data as folks transition over to this capsid. So all of that time, effort, and labor isn't wasted, but rather something that can be built upon to move forward to second-generation products for patients and communities. And we've made sure to put out our blueprints and roadmaps in public forums like this one, so that these slides are all available without confidentiality and without needing to have an agreement, so that not just patient advocacy groups and parents trying to progress programs, but also academics out there that want to be helping and want to be moving forward, but don't have the years and decades of experience, someone like Jim, on how to progress a program, can get that explainer guide, and so that we can be there to help them think through that. Thank you. With that, I think it's important to note as we progress through programs that there are going to be challenges and setbacks, and it's important to us as we do this that we are continuously transparent and we share those data and help make sure that everyone has access, because it is only through all of us learning together that we're able to progress this many programs. I have to have a huge thank you to Allison, the team, for this opportunity, and also Allison realizing these are my old slides where I had like 27 slides, and you said I wouldn't be able to get through them in time. So I don't want to go through all those and blow up your time frame, but I do want to just end on our thank you slide rather than talking through prion, which is really exciting, and come talk to me afterwards. But truly the thing that we're here for is to try to make sure there's not just a next generation of gene therapy, but of collaboration across the board in the rare disease space. One of my mentors said to me, it's not which disease I want to work on, it's the question of why not that one too. And I look forward to the day where more people are sharing that, and we're going to do everything we can in our power to make sure that this technology is available to foundations like FAST and many others. Thank you so much.
Thank you, Andrew. Thank you, Andrew. That was great. And I think everyone can see as we move things forward, the technology is changing, and we're very excited. We're going to hang on tight to see that first patient that's dosed hopefully in 2026 and how that can impact rare disease in general, particularly neurologic disorders.